I'm Jo Brewer. I'm the SVP for Allogeneic Research at Adaptimmune, and I'd like to introduce our poster today. So Adaptimmune is a world-leading biotech company in the treatment of solid tumours with adoptive T-cell therapy. In our current clinical trials, we engineer a patient's own cells to fight their cancer. We harvest a patient's T-cells and then put an engineered T-cell receptor into these cells that lets them see specific cancer targets. We then transfer these engineered T-cells back into the patient to kill their tumours. We call these cells SPEAR T-cells, and this type of treatment is called autologous cell therapy. Although we've demonstrated clinical efficacy using this approach, there are significant logistical challenges associated with making autologous T-cell products. There are many factors, including patient age, prior treatment regimens, or tumor type, that can affect the manufacturing of patient-derived products because of the variability in each patient's T-cells. Also, the time it takes to do this can be fairly long for a variety of reasons. This has led many people in the field, including Adaptimmune, to investigate the manufacture of T-cell therapies using a source that does not depend on getting cells from individual patients before manufacturing them. The goal here is to have a so-called off-the-shelf product. One way of doing this is to derive T-cells from stem cells. We are particularly interested in using induced pluripotent stem cells, or iPSC for short, to make our T-cells. iPSC are an attractive starting material as they have the potential to proliferate indefinitely in an undifferentiated, allowing large cell banks that could be used to produce multiple T-cell therapeutic doses. These types of stem cells are amenable to genetic engineering, allowing the introduction of transgenes or the generation of gene knockouts that could improve the T-cell function. T-cells differentiated from iPSC have been named iT-cells, and we'll be using that naming during our description of the poster. There have been several reports of T-cell differentiation from iPSC in the field, most of which have used stromal cell lines or artificial thymic organoids to support the generation of T-cells. Most cultures have also used human serum in their growth media. These extra components can be a source of contamination and potentially limit the ability to manufacture at scale for clinical use. Today, we'll show you our data demonstrating that we've been able to successfully generate T-cells from iPSC, and that we've done this without stromal cell lines or human serum. We have also been able to engineer in our enhanced T-cell receptors, and these engineered iT cells kill cancer targets in vitro. I'd like to introduce you to Garth Hamilton, who is the principal scientist on this project, and he's going to take you through the rest of the data.
Two approaches have been described for generating antigen-restricted iT cells. The first is the differentiation of iT cells from iPSC clones, which were transduced with the lentivirus encoding the TCR. The second involves isolation of antigen-restricted T cells and reprogramming into iPSC before differentiating back into T cells. We took an alternative approach and created a multicistronic fusion between our engineered T-cell receptor, or SPEAR TCR, and a gene that is expressed during the development of cells in the hematopoietic lineage. This allows co-expression of the TCR with the targeted gene. Unfortunately, we are unable to name the locus at this time. The work presented in this poster provides an update on the characterization and functionality of iT cells differentiated from iPSC that have the SPEAR TCR edited into them. For this study, we have used a SPEAR TCR that recognizes the MAGE-A4 cancer testis antigen, which is a protein expressed in several cancers. We clearly demonstrate that we can differentiate iT cells expressing the TCR that recognizes MAGE-A4 and that these cells are activated in an antigen-specific manner. We have successfully produced our own iPSC lines derived from the reprogramming of cord blood progenitor cells under GMP conditions. Figure 1 outlines the extensive characterization that was performed in each line. Genetic stability was assessed with numerous approaches, including karyology and cytosnip analysis to investigate gene copy number, losses or gains, and whole genome sequencing. iPSC pluripotency was confirmed by assessing expression of pluripotency markers including OCT4, NANOG, SOX2, and TRA160 by flow cytometry and immunohistochemistry. Additionally, RNA isolated from the iPSC was submitted for pluritest analysis. This is an in silico platform that verifies the pluripotent potential of iPSC lines, or a measure of their ability to make every possible cell type. For each cell line, we have a small cell bank of low passage number. In the experiments presented in this poster, we have edited the ADAPI-J line under research conditions to produce the clone 3D10, which has our MAGE-A4 SPEAR TCR knocked in. We also confirmed that these lines could be differentiated into T cells. Figure 2 summarizes our current process to make iT cells from these stem cell lines. Our differentiation protocol is a multi-stage process, and we can identify the emergence of defined progenitor populations along the differentiation pathway from iPSC to IT cells. As noted earlier, our process does not use serum or stromal lines for support. We seed iPSC colonies and promote differentiation towards the mesodermal lineage. As the differentiation in the colony progresses, we observe the emergence of complex 3D structures that are shown in Figure 2. Progenitor cells fill these vessel-like structures and eventually burst from them. Interestingly, these cells have early intracellular expression of genes associated with mature T cells, indicative of the potential to generate the cells that we want. We isolate progenitors from these structures and progress them through the remaining stages of the process. We can see the emergence of progenitor T cells, which further progress into more mature T cells while still maintaining the engineered TCR that we genetically edited into the iPSC line. So Figure 3 outlines additional phenotyping that we have performed on our IT cells using flow cytometry after differentiation. In the top row of flow plots, we have non-transduced IT cells produced from the ADAPIJ line. In the middle row, we have transduced IT cells that were transduced during the CD7-5 progenitor stage. And on the bottom row, in the red legend, we have IT cells produced from our edited 3D10 clone. These samples have been gated on CD45 lymphocytes, and the majority of the IT cells maintain the expression of T-cell markers CD7 and CD5. You can see that there are differences between the TCR edited cells compared to those expressing TCR from lentivirus or endogenous TCR gene rearrangements, as in the wild-type line. IT cells differentiated from the edited 3D10 clone are primarily double negative for CD4 and 8 or CD8 homodimer single positive, unlike the unedited cells, which primarily show CD4 and CD8 alpha beta homodimer staining. Although not shown, the edited cells will eventually transition to adopt a phenotype comparable to that of the edited IT cells. All the CD45 positive lymphocytes in the edited clone express CD3, as shown on the very right-hand panel of the flow plots, indicating that they have TCR expressed on their cell surface, unlike the unedited cells, where only half of the cells look like T cells, as determined by CD3 surface expression. The expression of CD56 is also higher in the IT cells produced from the edited 3D10 clones. The expression of CD56, coupled with this IT population being primarily double negative or expressing the CD8 alpha homodimer, suggests that IT cells differentiated from the edited clone 3D10 have a more innate-like phenotype, which has been previously described for IT cells differentiated from iPSC. In Figure 4, we look to examine TCR expression in these differentiated cells. So again, we have the same populations. We have wild-type. In Figure 4, we look to characterize the TCR expression in these differentiated IT cells. We have the same populations as in Figure 3. So on the left in green, we have IT cells produced from the wild-type ADAPIJ line. In the middle in blue, we have IT cells produced from the lentivirally transduced progenitors. And in the right-hand side of this figure in red, we have IT cells produced from the edited clone 3D10. So we looked for TCR expression in expanded IT populations by comparing total alpha-beta TCR expression with that of the SPEAR TCR that we edited in. We can detect the alpha chain of the SPEAR with an anti-V-alpha-24 antibody, and we can confirm the correct alpha and beta chain pairing by using the MAGE-A4 dextramer. As expected, the IT populations from wild-type ADAPIJ were negative for SPEAR TCR expression. SPEAR TCR was detected in a proportion of the lentivirally transduced IT cells. However, in the edited 3D10 IT cells, again on the right-hand panel of this figure, the TCR expression is relatively homogeneous with nearly all cells staining positive with both methods. These data not only indicate that edited IT cells express the TCR, they also imply that no other TCRs are present, and we are conducting further analysis to characterize the whole TCR alpha and beta chain repertoire to determine whether that is the case. This is an important point to understand for future clinical product development because uncharacterized TCRs have the potential to cause GVHD. In Figure 5, we show the results of assays where we have co-cultured our edited SPEAR TCR clones targeting MAGE-A4 with two cell lines. a melanoma line, A375, which naturally expresses MAGE-A4, and a colorectal line, COLO205, which does not. We then looked for several markers of T-cell differentiation, such as CD69 and CD25 by flow cytometry. We compared our edited cells to control cells, including peripheral blood cells transduced with the same SPEAR TCR, shown in the middle right panel. We compared several culture conditions. On treated COLO205 alone, COLO205 pulsed with a MAGE-A4 peptide, which should become antigenic because this cell line expresses HLA-O2, and A375 and A375 pulsed with peptide to see how far we could push the response. We show that activation can be induced in the HLA-A2 positive antigen negative cell lines by pulsing them for the appropriate MAGE-A4 peptide, and that the activation markers are expressed comparably to control cell lines derived from peripheral blood and transduced with the same SPEAR TCR. In short, the edited clones recognize antigen appropriately, as shown in the bottom left panel. They do not respond to target cells that don't have the antigen, and they do these things about as well as the positive peripheral blood controls on the right, and much better than the lentivirally conduced IT cells in the middle on the left. In Figure 6, we examine the production of cytokines and effector molecules associated with IT cell activation in these same conditions with the same test cells. We find that the edited clone in red were able to secrete interferon gamma, IL-2, and granzyme B in response to antigen. They did not do this when the target was absent. The profile was similar to control cells from peripheral blood, shown in purple, although the level of interferon gamma produced by the edited clone was slightly lower. In Figure 7, we performed killer assays using the target cell line, T2, that was pulsed with increasing amounts of the MAGE-A4 peptide. Again, we tested the same set of cells. We find that our edited clones, again in red on the right, killed target in a dose-dependent manner and performed even better than the control cells, shown in purple on the left. In Figure 8, we measure T-cell killing activity by IncuCyte assay. GFP positive A375, which are antigen positive, or GFP positive COLO205, which are antigen negative cells, have been co-cultured with the IT cells differentiated from clone 3D10. The growth of GFP positive tumor cells was followed using the IncuCyte live cell imaging platform. A decrease in green object count, or GFP, is a result of reduced cell proliferation and increased tumor death. And you can see in the lower curves where the SPEAR TCR expressing control or edited cells are co-cultured with antigen expressing targets, which are curves shown in red and purple. We find that our edited cells kill KID MAGE-A4 positive cancer cells as well or as better than the control transduced PBLs. In conclusion, the generation of TCR edited IT cells is an important milestone in producing an iPSC-derived allogeneic platform. The ability to promote TCR expression in IT cells via genetic knock-in at a single defined locus offers an opportunity to produce multiple clonal iPSC banks with engineered receptors to target a range of cancers. This off-the-shelf platform could provide multiple, defined and consistent cell therapies for people with cancer in a timely manner.